nomenclature of enzymes. Means naming of enzymes. Enzymes are named based on three different features, that is by adding suffix ase, ase to the term prefix. Here we can take example like prefix malt we can take plus ase, that is the suffix, together constitutes maltase. Next, another one, sucrose is the prefix or the substrate plus ase. We cannot call sucrase, we have to call it sucrase. So these both are considered as enzymes. Second, on the basis of depending upon the type of reaction that they are involved in. So second thing is basing on type of reaction. Example we can take pyruvate decarboxylase. See, during the linker reaction, means before entering of pyruvic acid into mitochondria, pyruvic acid is a 3-carbon compound. Because of its large size, it cannot enter into the inner membrane or into the mitochondrial membranes. So what it will do, it will remove one of the carbon dioxide molecule. There we can use the term like decarboxylation. In a link reaction we can write next. There another enzyme also we can include pyruvate dehydrogenase, because there is formation of NADH2. So pyruvate dehydrogenase. Here after CO2 is removed, it is getting converted into acetyl-CoA, which is easily diffused into the mitochondria for the next Krebs cycle reactions. Third one is based on the source. Example is bromelain or bromelain. These type of enzymes are obtained from the pineapple, because pineapple belongs to the family, that is Bromeliaceae. From pineapple, which belongs to family Bromeliaceae. So basing on these three different types of characteristic features and functions and sources, enzymes are named. Indiana University of Biochemistry has given some enzyme code numbers, which are also termed as enzyme commission numbers. So what are those? One is, it is considered as a type of classification we can call. Capital C, capital E, 1, 2. Again, capital C, capital E, 3. Capital E, capital C, 4. Capital E, capital C, 5. Sixth one, capital E, capital C, 6. Under code 1, enzyme code 1, the type of enzymes involved are oxidoreductases. Under enzyme code 2, enzymes involved are transferases. Under third one involved are hydrolases. Under fourth one involved are lyases. Under fifth one involved are isomerases. The last sixth type of code numbers involved are ligases. Now we will see one by one about oxidoreductases. These are also considered as 6 types of classifications, 6 types of enzymes given under classification of enzymes. So first one, oxidoreductases. The first one, oxidoreductases. This type of enzymes help both in oxidation and reduction reactions. Here we can take some examples like cytochrome oxidase. One enzyme we can take. Another one, phosphoglyceraldehyde dehydrogenase. This converts aldehyde form into acidic form. So here we can take one means a step where this enzyme is being involved. This is one example. This is second example. Glyceraldehyde 3-phosphate in presence of NAD plus and the enzyme phosphoglyceraldehyde 3-phosphate is getting converted into 3-phosphoglyceric acid. This is acidic form plus obtaining of NADH2. This is one of the example. Second one, transferases. Transferases means transfer of any functional group like amino group, carboxyl group, hydroxyl group, etc. One form of substrates are getting converted to other form. We will see one example also here. Transfer of functional groups converts one form of substrate to other. Example we can take here as glutamic acid, one type of amino acid which is having 5 carbons. This one plus pyruvate. These are getting converted into alanine type of amino acid and alpha ketogluteric acid. Now, one amino group from glutamic acid is shifted to pyruvic acid so that it is giving out new amino acid, that is alanine. One carbohydrate, because of addition of amino group, is getting converted into alanine. If you write the, actually here no need of biochemical structure, COO minus CH, C double bond O, CH3. If this is the pyruvic acid equation, structure, now it is changed into COO minus C, CH, NH2 and CH3. Means amino group has reached into the second carbon position. Now, pyruvate is 3 carbon compound, alanine is also 3 carbon compound, alpha ketogluteric acid is 5 carbon compound, that is other form of glutamic acid when amino group is removed. Third one, hydrolases. They break any bond by adding water. Example, starch. When we add water in presence of enzyme amylase, it gets converted into maltose, isomaltose and limited dextrin. They break bonds by adding water. Example, starch in presence of amylase or ptyalin. It gets converted into maltose, isomaltose, and maltose, isomaltose, and limited dextrin. Limited dextrins. Next, fourth one, lyases. Next, lyases also breaks the bond but without adding water molecule. Breaks bonds without adding water. Without adding water. Here we can take one example. Fructose 1,6-diphosphates in presence of enzyme aldolase gives one aldehyde form, one ketone form, that is glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate. Another example. Next, the fifth one is isomerases. Isomerases means they convert aldehyde forms to ketone and vice versa. Ketone forms to aldehyde forms. Convert aldehyde forms, or means one form to other form. Aldehyde forms to ketone or ketone forms to aldehyde forms. Or we can write vice versa also term here. Example, glyceraldehyde 3-phosphate in presence of enzyme isomerase are getting converted into dihydroxyacetone phosphate. The last one is about ligases. Ligases are also called as joining enzymes. Joining enzymes. They form the bond between two different or two fragments of the molecules. They form bond between two molecules. Two molecules, or we can also say two substrates. So here we can take pyruvic acid plus carbon dioxide in presence of enzyme pyruvate carboxylase. Carboxylase are getting converted into oxaloacetic acid. This is 3-carbon compound. This is 1-carbon compound getting converted to 4-carbon compound, that is oxaloacetic acid. So classification of enzymes based on protein and non-protein part. If any enzyme is made up of only proteins, then it is called as simple enzymes. So first one, simple enzymes. If only proteins behave as enzymes, example we can take as pepsin and trypsin. Then what about the conjugated enzymes? Conjugated enzymes have protein part and non-protein part also. Next, here includes protein part plus non-protein. There is a term that is, here only we will write. These two combinedly are called as holoenzymes. Protein and non-protein part. This we will write in an. So holoenzyme has two parts. One is protein part, that is called as apoenzyme and cofactor. Apoenzyme is considered as the protein part cofactor is considered as the non-protein part. Now, cofactor is again of two different types. That is organic part and inorganic part. Organic part and inorganic part, or inorganic types we can say. Here examples are all metallic ions. They constitute about 1/3 of an enzyme for complete activity of an enzyme. Under organic part, again it is classified into coenzyme and prosthetic group. Coenzyme is loosely bounded to protein part. Loosely bound to apoenzyme. Prosthetic group is tightly bounded to apoenzyme. Tightly bound to apoenzyme. We'll see just one or two points regarding coenzyme. Coenzyme is also called as inorganic catalyst. Inorganic catalyst. One point. Another one, here we can take, it is functional at high temperature, high pH, etc. Here examples under coenzymes are NAD+, next, and NAD, etc. These both are made of niacin. Niacin is considered as B complex, B3 vitamin. So we will write vitamin B3. Now, coenzyme is functional at a high temperature and pH. And high pH. Means basic conditions.